I am Sylvia Silver. I'm one of the directors of the CFAR Developmental Corps, and I wanted to welcome everyone. As you know, the main purpose of the CFAR Awards is to assist in increasing the amount of research in HIV that is done in a particular geographical area. And the Developmental Corps is charged with essentially running our pilot awards program, encouraging collaborations, mentoring, putting on research in progress seminars. And our journal club has been instituted for a few years now. We've gotten very high comments, I guess high marks from different groups on the innovation of our journal club. One, it is a webinar because we are a CFAR that is across the District of Columbia, and we all know how traffic is getting from one place to another, as well as the fact that it is not a regular type of journal club where a person will give a 40 to 45 minute presentation of science in a journal article. We have an advisory committee for the journal club, and the advisory committee a few years ago came up with the suggestion that we really foster collaboration by having one individual representing one of our cores in the CFAR. If all of you are not aware, we have not only the developmental core, but we have our social and behavioral science core, our clinical and population science core, and of course, our basic science core. So we choose one core each quarter to have someone present, not in a regular, very deep scientific way, an article that they found interesting from their point of view of their research, and to present it as sort of to a lay public so that people would be able to get an idea of what it is that the article is about. And then we have people responding from our two other cores to the article, again, from their point of view of what they think they could do in their area of research related to the article. So we then open it up to the public for discussion, the other members of the people on the webinar. So as you can see, this is not going to be a regular journal club. Most of you, as I see your names here, you've been to our journal club, so you know that this is how we do it. So we are very fortunate today to have Rachel Resop present an article in her area of research, which would be considered basic science. We have Blair Spence to be able to give us a perspective from the clinical and population science core. And we have a new member to our CFAR, Carlos Rodriguez-Diaz, who is going to respond from his area of research, which is the social and behavioral science core. So our normal operating procedure is that, Rachel, you will have about 10 to 15 minutes to give us an overview of what this article is all about and why it is interesting from your point of view. And then we will have each of the other two give about a 5 to 7 minute overview of where they are in their area of research. And then we'll open it up to the members on the webinar. So thank you, Rachel, for offering to do this. You are on.
Okay, thank you. Can everyone hear me?
Yeah, I would imagine so.
Okay, just checking before I launch into things. So thank you, Dr. Silver and the organizers, and thank you, Blair and Carlos, for agreeing to do this as well. So as was mentioned, I'm a basic science researcher here in MITM, and I'm also relatively new to the cure research field of HIV. Before this, I came from an immunology background. So this is actually really interesting to me. I'm still learning as well, so I'm really happy to take advantage of this and all of your expertise as I present this. So this is, as mentioned, this is a basic science article, but I will do my best to give a pretty basic overview. I did include the figures from the paper, but I'll pass over one or two of them just in the interest of time to try to keep things to 15 minutes or under. So if I happen to be going too in-depth or providing too cursory of a view, just let me know and we can stop at any point and just jump in with any insights. So this article is interesting to me because I'm currently working on a project looking at how cell-to-cell transmission and establishment of HIV latency are connected. So I found this article. It's a little bit older, but I think it's still interesting. So I'll just get started. The article is called HIV-1 infected CD4 positive T cells facilitate latent infection of resting CD4 positive T cells through cell-to-cell contact. So as most of us are probably aware working in this field, HIV is extremely challenging as a disease to try to cure because of the fact that it can become latent. And latent infection remains in cells in sort of a sleeping state and is actually the main barrier to a cure because it could be reactivated at some point in the future. And this makes it very challenging for us to study and to come up with treatments and prophylactics for HIV. It's been known in our field that cell-to-cell contacts promote infection by a variety of mechanisms, one of which is the formation of virological synapses, which is basically the concentration of a large amount of virus at cell-to-cell contact sites. Contact between cells can also help to overcome barriers to infection, both cellular and environmental, for example, antiviral factors, neutralizing antibodies, and poor fitness of virus. So I believe that it's important to better understand the role of cell-to-cell transmission in how HIV latency is actually established. So again, this is pretty basic, but how it's actually established within cells. And this may influence cure strategies or responsiveness to ART, for example. Sorry, just getting used to how the slides change. So these researchers were interested in developing a model to better understand the process of cell-to-cell transmission. So very basically what they did was they infected activated CD4 positive T cells by a method called spinoculation, spinning with virus, for 48 to 72 hours. They then co-cultured with autologous resting CD4 positive cells. So these were resting T cells that they isolated from the same donors. This is all done in primary donors. And then they cultured for additional 72 hours. But they did administer sakinavir after 24 hours. This is an antiretroviral. So this allowed them to look at the events that were happening within the first 24 hours in their model. They then did several downstream analyses. They separated the targets to look by a PCR analysis of integrated HIV DNA. And they also looked at reactivation of the virus that had been the latent virus in the cells. And they did this by reactivating with a stimulus and then adding some cytokines, IL-2 and 7, which are important for T cells, and analyzing by flow cytometry and again by PCR. So that's their general model. Their first main question was to determine whether activated infected CD4 cells transfer virus in co-culture. So it's not necessary to get into the nitty-gritty of their flow cytometry gating scheme, but what they did here is they looked by a method called flow cytometry to examine different cellular populations. And they selected very carefully live cells and their resting CD4 targets. And then they also included markers to look at cellular activation. So they were able to gate on cells that were not activated using these different markers. And their first observation was that by this PCR technique, which examines integration of virus after co-culture, they were able to see integrated HIV provirus in the cultures in their model after co-culture with activated infected cells. Interestingly, they were able to see this with both X4 and R5 tropic HIV molecular clones. So this is HIV used in the lab that can use either of the two well-established co-receptors for HIV. So this is the figure showing that this is their antiviral control where they didn't observe anything, and then with both X4 and R5 tropic HIV in their infections, they saw integrated provirus by this PCR technique. They then wanted to know whether the cells were actually activated in their model. So again, they looked at CD69, HLA-DR, and CD25, 3 different activation markers, and they compared before co-culture and after up to from 1 to 4 days. And they were able to see that in their co-culture model, there is a weak but statistically significant increase in these markers of activation. So from that, they thought that there was not a robust activation, but there was likely some weak cellular signaling going on in the transfer of virus from infected activated cells to the resting cells in their model. Their next question was to determine whether the actual physical contact of cells was required for transfer of virus. So to do this, they utilized a method called Transwell assays. And this is actually a pretty neat technique where you can separate different populations of cells using a sort of cheesecloth membrane, as you can see diagrammed here. I'm not sure if my pointer actually appears on the presentation, but they used a Transwell assay to separate their activated and infected cells and then their target cells, which were the resting cells that you could see in the first figure that they separated out. They compared that to the co-culture that they had done before, and they looked at whether there was actually integrated provirus in co-cultures versus cell-free separated cultures. And again, when compared to their antiretroviral treated control, which should be negative or low, they saw that there was a statistically significant increase in integrated virus in the regular co-culture where the cells were allowed to touch. But when the cells were separated by the membrane, there wasn't an integrated virus detected above a certain level, above their background. So that led them to believe that it was actually really important to have the direct physical contact of the cells in their model. I'll just go really quickly over this. This is not important, as important, but they wanted to ensure that the virus was actually passing through the membrane from the activated infected cells. So they plated the TCMVI reporter, HIV reporter cells in the lower well, and they just allowed the virus to fall through. And they saw quite a bit of luminescence, luciferase, from that reporter cell line. So they knew that the virus was actually getting through. But there was something actually important about the direct contact of the cells from what they were seeing. They then went on to do several different types of microscopy, both live cell imaging and confocal microscopy, which I'll show a couple of examples of in the next slides. But just to give for those who are interested in the more molecular background, this is the construct that they use. This is an NL43 GFP GAG. So this is a fluorescent HIV. So you can see here that they started to image some different cellular contacts. And they could see an activated infected cell, which is larger, and it's fluorescing green. And then the smaller blue cells are labeled with a cell tracking dye. These are the resting cells, and CD4 is in red here. So you can see that they started to, when they did microscopy, they could observe actual cell-to-cell contacts and a concentration of virus. And they did observe greater GFP fluorescence when cells were actually in contact. So then they did a little bit more microscopy to get deeper into exactly the nature of the interactions. Again, just to mention, they did they had a GFP producing NL43 and CMAC, which was their cell tracker blue dye for the resting CD4 cells. I want to just go to the next slide because it's a little more interesting. You can actually see video of transfer if it will play. I'm not sure if it will play. It doesn't look like it's going to play here, which is unfortunate, but if you're interested, you can go to the actual article online and you can just click on the video right there, and you can actually see some really neat interactions between the larger infected CD4 positive cells with all this virus and the resting cells. And this one actually, which is on the far right, I would recommend you go watch that one. It's actually pretty neat. You can see a GFP fluorescing cell come in and kind of engulf and dance over the resting CD4 cells. So they observed a variety of different interactions. They observed polarization of HIV, which they could see kind of clumping into certain areas of contact with the resting CD4 cells. They could observe both types of cells simply carrying virus particles, and they could observe a transfer in a variety of ways. So it was actually some pretty neat microscopy. And this is just another example of the different types of interactions that were observed, if you're interested to see further. So then the last bit of data that I like to just show before kind of getting into the discussion is that they were finally interested in determining whether there was a difference in the inducibility of the latent virus, depending on the means of transmission. So we're all interested in how to induce latent virus, which means to get the cells that have virus latently infected to start producing that virus so we can detect the cells and kill them and eliminate the virus. So they wanted to know, is the latent infection, which is established in their model, actually inducible? So what they did was they actually, something similar to what we're doing here in the Bosque lab, they reactivated the virus from the resting CD4 cells with IL-2, IL-7, and CD328, which is a stimulus which we actually use as a positive control for viral reactivation. And they were able to determine, first of all, that in their co-culture, this is just compared to the unstimulated control, they were able to determine that there was activated HIV from those latently infected cells. So this was reactivation observed, was about 1% average, and that's pretty consistent also with what we see sometimes. So that was kind of interesting to me. So they wanted to compare, okay, so we see reactivation. What about when we compare that to other methods of infection? So cell-free infection or spinoculation, which is a little bit more robust means of infection where you spin with a large amount of virus. So they compared first to spinoculation, and they saw that when they reactivated those cells after infection, there was a little bit higher of a reactivation. When they compared to a cell-free infection, in which they just used a concentrated amount of NL43, but they didn't spin, they saw a little bit less robust of reactivation, actually not really much reactivation at all in a couple of the donors in that experiment. So what they did was they calculated the inducibility of the virus by taking the percent GAG reactivated. So this is by flow cytometry, the P24 that was reactivated, divided by the provirus per cell, which was the integrated virus determined earlier in their studies that you saw in the model in the earlier figures. And by taking that equation, they determined that their co-culture condition was on average about 21% inducible, whereas the spin infection was about 43% inducible, and the infection without the spin was more intermediate, about 31% inducible. And of note, they didn't have a huge amount of donors for these conditions, but it kind of gave them an idea of how their co-culture model compared with reactivation to other mechanisms of infection. And here you can just see that summarized in figures D and E. You can see by infection type, comparing their control, again, the antiretroviral, to co-culture, spin infection, and cell-free. And then you can see they're comparing the ratios of reactivation, which is actually the more important information. So how much of the virus that is integrated can be reactivated. And I think this is actually probably, for me, the most interesting, apart from the really pretty microscopy, the most interesting takeaway from this paper, that this is good preliminary data, and others have and are working on this as well. This is good data to show that the inducibility of the provirus might actually, is likely in part related to the means in which the latent infection is first established. So that seemed to be pretty important to me. So just to summarize and try to start a little bit of a discussion, these authors found with their model that cell-to-cell contact is an important means of transmission. They do induce latent infection with this model, and the latent virus may be more resistant to reactivation when compared to cell-free infection. But again, a major caveat is that was a spin infection that they did, one of their two comparisons, which is not exactly a very physiological means of infection. So just to keep that in mind. And also, reversing latent proviruses may reflect the manner in which the infection is initially established, which I think is pretty interesting. Now, some of this has been shown before. It's been shown that resting cells isolated from tonsils contain HIV that can be reactivated. And also, it's been shown and is currently being investigated that the mechanism of entry can affect the reactivation of latent virus. And also, there are other groups doing this. I had edited this slide, but it wasn't in time because I had sent them already yesterday. So there are other groups working on this as well that I haven't credited. And also, I feel that their model doesn't accurately describe resting cells, true resting cells, because during their co-culture, they did see that they're weakly activated. And also, I'm not 100% sure that these are perhaps they could have done different markers as well. And it seems that CD69 may be the only of these three markers that would have really changed significantly during the time point that they were looking at. But that's debatable, and others may have more expertise in this area. But I do think that these cells weren't truly resting by the point that they were examining them. So they may have been in more of a slightly activated or primed state due to that contact with the activated CD4 cells. And this is actually a really interesting sort of middle ground, which I am not sure is well characterized. But again, I would welcome anyone's expertise on this. So most of all, I would be really interested in seeing more from this group to actually examine the mechanism of the difference in inducibility by the different means of infection. So hopefully this is something that they may continue to work on in the future. But I think this paper is actually really interesting for us basic science researchers. I was a little bit more challenged to relate this to behavioral and social science research. So I'm sorry if I did select a paper which might have been a little bit more challenging to relate to these other areas, but hopefully everyone was able to get a little bit out of it. And thank you for listening, and I'm looking forward to a discussion.
Very good. Thank you very much, Rachel. Okay, as is our custom now, we're going to go to Blair, who represents the Clinical and Population Science Core, for her take on this article.
Hi, everyone. This is Blair Spence from Georgetown. I will be representing the Clinical and Population Science Core. So actually, my main interest is in HIV-related comorbidities and end-organ disease related to antiretroviral therapy. So this isn't really quite in my wheelhouse, but I actually thought it was a very interesting article, and I really liked the way that Rachel reviewed the article, because I actually had some additional thoughts now that I heard Rachel go through the article. Because initially I was struggling a bit to kind of come up with a direct kind of clinical a way to apply this clinically, or use clinical consequences, or how this applies to my research. But I did come up with some, I think, interesting questions that I thought. So, I'm not a basic scientist, so I can't comment on, the methodology that the authors used for this analysis. But initially my thought was, just kind of looking at this from, a 10,000-foot view, from a clinical perspective at least, and, for the population of those living with HIV as a whole, right now we have, effective therapy, and the goal is, viral suppression with the therapy. But, ultimately the ultimate goal is cure. And at least from my understanding of the article, it does leave kind of a deeper understanding, or at least a gateway to additional studies that can be done to understand viral latency, as well as to look at how inducibility of virus. And I think that has direct sorry, my words are coming out a little rough today. It can be directly applied to cure research, which is kind of, our ultimate goal. But I guess I do have kind of a general question, either for Rachel or for the group. I was really struck by, I think, Rachel, you mentioned that you thought this was the most interesting part of the article as well, is that, only 21% of those from latently infected had inducibility. So how much of that population actually is made up in the latent reservoir? Sorry if I didn't phrase that quite correctly. Are you still there?
Yeah, Rachel, did you hear the question?
I sort of did. It's a little bit fuzzy on my end. Sorry. The question was,
of the 21% that are latently infective, how many of them actually can be induced? Is that right, Blair?
No, because my understanding is that there was the cold culture generated latent virus was more difficult to induce, right? And you said 21% were inducible.
Mm-hmm.
But how much of that is made up in the population, or is that heterogeneous depending on the person?
Oh,
Or is it
is the question how much of that difficult to induce virus is a part of the latent reservoir overall?
correct? Yes.
Oh, that's a great question, and I'm actually not sure the answer to that, and I'm not sure that anybody is 100% sure.
Anyone knows the answer? I guess it's
This is Alberto. I may comment on that. I mean, it is known that even in patients, there is a part of the latent reservoir that is not inducible. And I mean, it is intact because like one of the differences between probably in this in vitro system and what happens in vivo is that latency in vivo, there is a lot of viral sequences that are integrated into the genome that are defective. So that doesn't produce any viral product, doesn't produce infectious variants, but they are there in here. I think we know now from studies from Silicano that there is an stochastic reactivation of latent virus even in the presence of a maximal stimulation. That is what they use in here. So you will have some viruses that are reactivated, but some viruses that are latent and the cells get activated, they don't get reactivated. And the mechanisms behind that is what I don't think that is known.
Oh, I see.
Do I answer the question?
Yes, thank you.
Anything else, Blair, that you wanted to add?
No, I think that was all I had to add.
Okay. So all of the people that you're treating with antiretrovirals, all of this is going on in their body at the same time, correct?
Correct, yes.
Yeah. Okay, thank you. Carlos, your comments?
Hi. So, yes, thanks for the invitation, Dr. Silver. My name is Carlos Rodriguez Diaz. I recently joined GW in September last year. I'm an associate professor in the School of Public Health. My background is community health and health education. I don't have any training in basic or clinical sciences, but the discussion has been very helpful to frame some of the thoughts that I had after reading the article. I have to agree with a comment made by Dr. Resop on how the latent infection is established was part of what I understood as an interesting angle to translate all these findings into working with people and their environment. Definitely, this article is all about the cells and the cell environment. And I am taking this to my realm of action, which is working with people, their behavior, practices, and the social environments. So there are three areas that have some further ideas and thoughts. One is related to what puts people at risk. So here we are talking about latent infection and cell-to-cell transmission. And that made me think of how in social and behavioral research and somehow in clinical practice, we have defined what is considered risk. Actually, for transmission of the virus, we only need one risk practice. However, all the frameworks and the research that we do use very ambiguous terms to define the risks that people may have for HIV infections. I will use as an example the word promiscuous. What that specifically means is very relative. How many sexual partners do you have to have in order to be considered promiscuous? If we look at the literature, it's not very consistent. Therefore, as there are many ways of transmission of the virus, but we have certain amount of ways of preventing, that was very thought-provoking to consider how that we've been framing risk and transmission. Also, another angle was how is that we can protect individuals for infection. We have used risk models. Right now, we are considering a new plan to end the epidemic in the U.S. However, one of the gaps that I have identified in that plan is HIV testing. So actually knowing who is infected. This article invited me to think about those who are people who are with HIV and have no knowledge of their infection. particularly those who are acute infections. And the models that we have now is to look for those that are at increased risk for infection, but we are not necessarily even considering those that may have less risk for infection, but are actually engaging in the practices that are conducive to HIV transmission. So it's an invitation to think where we should focus and strengthen our prevention efforts. And finally, and connected to that, and it's something highlighted here in the article, on the different biomedical approaches that are considered in the process of infecting a cell, made me think about the biomedical approaches that we have to prevent. There is a lot of focus right now on PrEP, but this article made me think of PEP and the needs that we have to make PEP available to populations that are at risk for infection, and also to people who might not be at increased risk of infection, but that can identify when a risky event happened, and it's a resource that should be readily available for them, and it's not. And finally, and more from the community health, communications health perspective, I should say, sorry, this article invited me to think about the implications of messaging around U equal U and undetectable equal to untransmittable. Because in fact, that requires a sustained undetectability in order to avoid or promote certain practices that will look like as latency and eventually can increase personal and community risk. So definitely there is some work that we can do in order to provide the resources needed for those who are with HIV to remain HIV undetectable, and also to reduce the risk of new infections, other STIs or other infections overall, and reinfection of the virus. So I hope this is not a stretch of the idea behind the concept for this journal club. Certainly as a non-basic scientist, it was an adventure to read this and make sense of all the components, but it was in fact very interesting to take it to my realm of action, and hopefully my thoughts are conducted to good discussions next.
Thank you, Carlos. It was very interesting, and I'm happy to hear that we may have sparked some new ideas. All right, well, as we have said, we are now open for discussion. Is there anyone that has a question or comment or would just comments? Please feel free.
This is Michael. I actually wanted to ask Rachel about her kind of interpretation of the implications from this paper. So basically, the finding that infection from cell-to-cell contact is more effective than from infection by cell-free virus is a node kind of story, right? So basically, and some people even believe that there is no free virus infection of T cells taking place at all, that most infection occurs through cell-to-cell contact. Now, what they show is that this cell-to-cell contact is more effective in establishing latent infection, right? That's what they claim. But on the other hand, this latent infection that is established is less likely to be activated by stimuli. So my question is, what is new about how does this move our understanding of latency in general? And if it does it, I mean, in my view, it doesn't add anything.
I actually agree with you on that, Dr. Bekrinsky. After I read the paper and I was able to get a little bit more into the literature, which I'm still learning, I did find those other studies that I mentioned, as well as some others who had basically shown this before. I think the reason that this article may have been published where it was, and and it came out when it did is possibly because of the different microscopy images, which were pretty nice in my opinion, which kind of showed things in a different way. And the model is slightly different from other models that have been used before. But apart from that, I actually agree with you that this doesn't really show anything we didn't know, for those of you who knew the literature, or really move the field forward in an entirely novel way. However, I thought it was actually interesting to kind of give a good overview of latency for all of us, those of us who are still learning and those who are not basic scientists. So that's why I chose the paper.
Yeah, no, I'm not questioning your choice. I think it's a really very interesting paper, and basically it gives kind of some ideas of how this process takes place. But I was wondering whether there is any implications for our understanding of the biology of latency, and I couldn't really find any. I mean, the fact that cells. I mean, the only thing that I could think of is basically where does this contact between activated infected cell and the latent cell occurs, and obviously this can only occur in lymphoid tissue, right, and in specific areas of lymphoid tissue. So that's where basically latency is established. But that we knew before as well. So I just think that biologically there is not much new that we'll learn from this paper. But I mean, I agree with you that this movie is cool and basically it's really very impressive.
Yeah, I think that was kind of the reason that the paper kind of came out in Cell Reports. But no, I agree with you that I would really like to, as I mentioned, I'd really like to see more in-depth work into the mechanism at this point, because this is kind of known. So now I'm interested in, okay, so they have these techniques, these microscopy techniques established. How about imaging some of the other components that may be involved with the membranes and looking at the polarization, the assembly of lipid rods and things like this? That would be really interesting to see.
So I have a question. Unless someone else in the audience has a comment before, I don't want to be hogging as moderator. Does anyone else want to ask a question or make a comment?
I want to make a comment about to Michael about. I mean, I do think that it has some value to know that how HIV established latency or the mechanism may impact how the virus reactivates, and mostly for cure approaches and latency reversal agents, that it seems that obviously during spin infections there is a lot of changes in the cellular environment that happens, and that may change how the virus actually infects the cells. So it kind of, although it was known that cell-to-cell transmission happens, I think it gives us a little bit of more in-depth, or we can, and I agree with Rachel that I will have like more mechanistics of why it's less inducible, but that actually, it opens the question of in vivo and why you have viruses that cannot be induced from individuals, or is this model be used, or similar models, to understand what is the difference between a virus that it can be induced and a virus that it cannot induce, because that will have implications on cure approaches. So I think it has some value. It's in that regard. I don't think that people know yet what makes a difference into a virus that is inducible and a virus that is not inducible. Still, they don't address that, but I think the model may be a suitable model to address those questions.
Anyone else? Any comments? Well, I guess I was wondering whether there was anything that was that when we think about other areas of research, is there ever any thought about trying to get some of this knowledge? Of course, we're always trying to prevent HIV infection, but is there any indication from any of this in this article that could be put out to the public in some way to further explain what is happening and why we need to make sure that PrEP is used or people are trying to not get infected? Is there any message that can be brought to the public's level? And I'm not saying that in any derogatory way. I think you all know what I mean. Does anyone see anything that could be going farther from this article to help any kind of research related to the public or to our patients?
Yeah, this is Michael again. I'm sorry if I just want to comment about Sylvia's question. So I believe that what this paper teaches us, or at least what we can learn from this, with regards to this potential establishment of viral reservoir and things like that, is that basically it's very dangerous to have activated infected cells. So that's, I would say, the bottom line. And for people who take drugs and have suppressed virus, this is really important to realize that all these so-called viral leaps, or whatever you call them, when you have this kind of appearance of virus periodically, that these are very dangerous, because these events actually cause amplification of viral reservoir through this cell-to-cell contact in lymphoid tissue and things like that. I mean, this is not something new that they discovered, but it kind of reinforces the previously known previous understanding that that's how this reservoir is maintained and sometimes amplified, but at least it doesn't really go away due to continuous replenishment from such events. So basically what is important for infected people to know, and what can be really told them in public appearances by CFAR members, or I mean in any presentations, is that it's extremely important to keep viral replication to the minimum, to avoid those appearance of those infected activated cells. So I think that's the value of this paper for this kind of prevention and HIV maintenance field.
Thank you, Michael. Does anyone have any ideas of how we could achieve that? Or any research we could do into thinking about how we could find a way to achieve that? Well, all right. Very quiet group this time. I'd like to thank Rachel for bringing us this article, and Carlos and Blair for responding to it. As you know, our journal club is given once each quarter. And so if anyone on the call from the social and behavioral side or the clinical and population side would like to present their article that they found interesting at our next journal club, which will be sometime in the spring, please contact me. If not, I'll be contacting people, because this is a CFAR program. We're very excited about offering it this way, and the webinar will be available after we get off. One last chance. Anyone have any comment or anything? Well, again, thank you to our presenters, and thank you for everyone for joining us, and have a great day. Bye.
Bye.
Thank you very much.
Thank you.
Bye.
Thank you.
Thank you, everybody.